Hello, my name is Panda Connor, and in this video, I will be discussing the genetics behind the circadian clock. We will first start with the definition of the circadian clock, then move into an introduction and background into the three scientists who discovered the gene sequencing. Next, we will discuss the proteins and genes involved in this process and the mechanisms at play. Then we will look at how our knowledge of the circadian rhythm affects our society. Finally, all the sources used will be listed on the screen. I'm sure most of us have heard of the circadian rhythm. I would presume most of us know it as a mechanism that makes us sleep at night and keeps us awake during the day. Let's first break down the word circadian. Circa means around and dies means day. Now let's talk more about the circadian clock mechanism and how it relates to our bodies. The circadian clock does more than just make us sleepy. It also plays roles in our behavior, hormone levels, body temperature, and metabolism. As seen in the provided visual, at 6 p.m., we tend to have the highest body temperature. Similarly, at 6 a.m., we have a large cortisol release. Dr. Jeffrey C. Hall was born on May 3rd, 1945, in Brooklyn, New York. He grew up in Washington, D.C., and began his studies at Amherst College in Massachusetts. He then changed his focus to genetics and studied at the University of Washington in Seattle. He earned his PhD in 1971. He continued postdoctoral studies at the California Institute of Technology. From there, he went on to become an assistant professor of biology at Brandeis University in Massachusetts. At Brandeis University, he met Dr. Michael Rosbash. Together, they used Drosophila melanogaster as their model organism, looking at the genes and proteins involved in the circadian clock. In 2009, he received the Gruber Prize in Neuroscience. In 2012, he received the Canada Gardner International Award. Finally, in 2017, Dr. Hall, along with Dr. Rosbash and Young, received the Nobel Prize in Physiology or Medicine. He continued to teach until 2012. Dr. Michael Rosbash was born on March 7, 1944, in Kansas City, Missouri. He was raised in Boston and studied chemistry for undergrad at the University Institute of Technology. He got his bachelor's degree in 1965. He went on to study biophysics at the Massachusetts Institute of Technology, graduating with a PhD in 1971. He then became an assistant professor at Brandeis University in 1974. At Brandeis University, he met Dr. Hall. Together, they isolated and sequenced the period gene involved in the circadian clock cycle. Dr. Rosbash won the Gruber Prize in Neuroscience in 2009, the Louisa Gross Horowitz Prize for Biology or Biochemistry in 2011, and the Wiley Prize in Biomedical Studies in 2013. Finally, in 2017, he was awarded the Nobel Prize in Physiology or Medicine with Dr. Young and Dr. Hall. Dr. Michael W. Young was born on March 28, 1949, in Miami, Florida. He was raised in Miami and attended the University of Texas in Austin, where he got his bachelor's degree in biology in 1971. He continued studying there and was awarded his doctorate in genetics in 1975. He then finished his postdoctoral studies at Stanford University School of Medicine. Next, he joined Rockefeller University as an assistant professor and became a full professor in 1988. At the same time as Dr. Hall and Dr. Rosbash, Dr. Young isolated and sequenced the period gene. Additionally, Young found that introducing a fragment of DNA from the period gene loci into a mutated cell where the circadian clock was disrupted restored the proper circadian rhythm. This demonstrated the functional significance of the period gene. Dr. Young also identified the gene double time, which we will discuss in just a few slides. He was awarded the Gruber Foundation Neuroscience Prize in 2009, the Louisa Gross Horowitz Prize in 2011, and the Canada Gardner International Award in 2012. Additionally, he received the Nobel Prize in Physiology or Medicine in 2017. You probably noticed a trend with the awards. Each scientist was awarded the same awards as they continued to work on the same research projects together. Our understanding of the circadian clock began in 1729, when Jean-Jacques de Ortiz de Marin showed that mimosa leaves open at dawn and close at dusk. He hypothesized that this phenomenon was due to the presence of light. Next, in the 1970s, Seymour Benzer and Ronald Konopka at the California Institute of Technology created mutant flies with abnormal biological clocks by manipulating the same gene. They decided to call this the period gene. Finally, in 1984, Rosbash, Hall, and Young were able to isolate and sequence the period gene. Rosbash showed that PER protein rose and fell over 24 hours, peaking at night. He thought that PER interferes with the period gene, creating the biological clock. At the same time, Young figured out how PER got into the nucleus. Another gene, timeless, TIM for short, allowed PER to enter the nucleus and stop period from making more. The circadian clock mechanism works in a negative feedback loop. The period gene codes for the PER protein. Timeless, also known as TIM, is important as it binds to PER and brings it into the nucleus. Additionally, genes clock, CLK, and cycle, CYC, bind to the enhancer box of PER and TIM, creating positive transcription factors. This activates transcription in the cytoplasm. TIM then brings PER into the nucleus, where PER turns off its own transcription. This is a negative feedback loop, preventing further synthesis of itself. This mechanism is in place to ensure that the concentration of PER in the nucleus does not become too high. Throughout the afternoon and into the evening, transcription of PER is activated, allowing PER's levels to begin building in the cell. This buildup is what causes us to become tired by the late evening. Once there is enough buildup, the PER transcription stops and PER degrades in the nucleus. To find and isolate the period gene, the researchers cloned the gene from the fruit fly models. Also of note, Dr. Young discovered the gene double time, which encodes a protein that helps regulate the frequency of the PER oscillations over a 24-hour period. Young, Hall, and Robach's work gave an amazing head start into being able to understand our bodies so much better. After all of this, you may be wondering why any of this is important. As mentioned at the beginning of our presentation, our circadian rhythm regulates more than just our sleep, although that is a big part of it. It regulates other body processes such as metabolism. It is important that we understand how our bodies prefer to operate and we listen to them. Not sleeping the correct amount of time or at the right time leads people to be more at risk for disease, as well as being more accident prone to not great sleep, which can be dangerous. Two known circadian clock disorders that affect people's sleep are familial advanced sleep phase syndrome and delayed sleep phase syndrome. In advanced sleep phase syndrome, those that are affected get extremely tired in the early evening, such as around 7 p.m., then wake up on their own well before dusk. In contrast, delayed sleep phase syndrome affects people in that they do not get tired until very late at night, and thus do not naturally wake up until the late morning or early afternoon. Additionally, their circadian rhythm has implications for other disorders, such as seasonal affective disorder, SAD. Patients with SAD are impacted by how much light they receive. During late fall and throughout winter, there is less sunlight during the day. This can cause an onset or worsening of depressive symptoms. Because the circadian clock is also affected by light availability, it is thought that the circadian rhythm regulating the body's processes contribute to SAD. Here are my cited sources. Thank you for your time.